Today I will try to show you some of the typical work that we are doing in genetics and genomics of mental disorders, and I present you a very simple schematic representation of our current model. And our current model actually works on one particular framework that we call brain imaging genetics, where what we're trying to do is just mixing up together information from what we can get from brain imaging together with information that we can get from genetics and genomics. And Hal, in the first part of the afternoon today, has already presented one example of imaging genetics, or at least brain imaging, where he showed how we can move forward in comparing different subjects. The principle is very simple, and what we think to do is that we know that we can move from genes to cells and systems. So genes are the basic building blocks for whatever kind of molecules that we can have in a cell. In our case, a neuron. And then what we can know, what we know is that cells are building up systems. In our case, the brain. What we think is that we can then imagine that there will be any kind of possible relationship between the brain and the behavior. Okay, so that is the basic of our brain imaging genetics, where instead of studying a simple disorder, then we study a brain imaging function or dysfunction in a given set of patients. The problem is that we don't have one gene or one cell, but we have a multiple event. We have multiple genes, and then we have several different types of cells and subcell component. So in essence, every single object is not one object per se, but is a network of different objects. And the relationship between a cell and the basic building blocks of genes is not a one-to-one, but is many-to-many, because we have many genes that are building up a different type of cells or cell components, then are building up different part of one system, which is in this case the brain system, and so on and so forth, up to the symptoms and the disease. So the complexity is actually there, and another important point for our talk today is that the cost of sequencing is just reducing to almost a negligible factor in the terms of science production, because if you just see and if you remember how much it cost our first genome sequence, which was $2 billion, and the second genome was $100 million. Now we are at each single genome sequenced in a small lab like my lab cost approximately $1,400. Okay, so what is happening is that with a reduction in cost, we are incredibly increasing the amount of data volume production. And the amount of data volume production is translated into an enormous amount for what is related to my interest in what we're doing here, an enormous number of genomes sequenced. So if you're just trying to combine the incredible number of genomes that are already being sequenced and that will be sequenced in the next years, together with all the information that is already available or is available in our patients for whole brain imagings, then we are talking about petabytes of data set, even for a relatively small lab or for one consortium like our own consortium in Southern California. So what we are facing is now a deluge and flowing of data that are rapidly reaching the petabytes volume, and then what we need to do is just to analyze all this kind of data, trying to make some kind of inferences and try to find out some kind of interesting solutions or interesting ideas about how the system works and how diseases might be generated in a dysfunctional system. So the demand of the user is to have a system which is relatively easy to use, but we have seen from talks before that everything that looks easy actually is not easy at all. Okay, but then we need to find out some system that allow us to make some kind of work with the final object that we're interested in. So just to give you a few examples, and then I will just move into the final part of my talk. When we are talking about the brain, we are in this particular situation here where what we want to do is analyzing the human brain, okay? But then analyzing the human brain per se does not have any sense unless we know what are the different parts of the brain that we are interested in, and essentially we don't know which are the parts of the brain that we are interested in a priori, unless in very few special cases. So what we need to do is just going through a complicated algorithm and trying to generate all this kind of information about different parts of the brain that we can then compare and combine together. So partitioning and segmenting the structure of the brain is one of the key features for this kind of work that we are doing particularly right now. The other component is the amount of sequencing. And so today sequencing is extremely low cost, as I said, but then the data that are produced are about billions of data points for billions of nucleotide information per each single genomes, okay? So again, the only way that we have here out on how to do this kind of analysis for this particular type of information is to use workflows or to use algorithms that allow us to do a semi-automated analysis of all the sequences data. And just to give you an idea, anytime that I am doing a sequencing, I am trying to sequence 4.5 million nucleotides in our cells, but then I ended up having an excess of 10 to 30 times that number, just to be sure that I'm not doing any kind of mistake. So for any single individual that I wanted to sequence, I ended up having something like 120 billion data points to look at, okay? So, and again, what we get at the end of the sequencing process is something like that, which is totally not understandable for anybody, not even for a psychiatrist, okay? So, but what we essentially have is that you can just have a small fragment of your genome, and then you have to assemble each of these small fragments into a blueprint of the human genome, as we know, and just making the references about what we know about the genes, what we know about the variants of the genome, and what we know about the complexity of the genome. But the complexity of the genome is way much higher than we are thinking in terms of just the pure sequence. And the sequence is just the linear combination of nucleotides, because when we go into the analysis of the human genome and the human genome sequences, we have to take into consideration the complexity of the genome. And the complexity of the genome is more or less summarized in this particular slide, where we know that we have genes that are coding. Thank you. We have variants that are single nucleotide polymorphisms, and then we have functional regulations in terms of whatever happens around the genes that regulate the expression and then the function of that genes. So this is what we knew, but what we know today is that we have an enormous amount of information that was hidden in the genome that we were not aware of, because we know a lot about how not only genes but also non-coding portion of our genome got transcribed in RNA, and we know a lot about all the different types of set of regulators that are too many to discuss, but just to give you an idea of the complexity that we're looking at. So when we try to merge together the complexity of the genome with the complexity of the brain image and try to understand something of the brain, it looks like a difficult task, and actually what we are facing is a lot of challenges in brain imaging genomics, not big data only, but brain imaging genomics. And some of the challenges are very simple. So we are simply facing thousands of sequenced genomes as of today, okay? So our small consortium in California have just sequenced 1,000 genomes of schizophrenic subjects in controls, and we are now expanding to some sort of an exponential volume. And just for autism, there are 18,000 sequenced genomes available, okay? And yes, it is feasible, it is easy to get into the internet and download this kind of stuff and doing the analysis on your own computer, but 1,000 genomes are about 80 terabytes of data. Are you sure that we can really have all 80 terabytes of storage data to download whatever we want on our computers? Maybe yes, maybe not. And then it's not only that, because this is just the storage volume. If you want to work on, I think it's okay, if you want to work on one genome, the genome explodes and give you zillions of tiny files that are the nightmare of Harry Mangalam here, and explode in about 1 terabyte of data. But then there are even more complicated issues, like the human genome got updated 3 times per year. If you really want to follow up the complexity of the annotation, you have to redo your calculation at least 3 times per year. Okay, we have 4.5 million variants in 1,000 subjects. It's 4.5 billion variants that we have to take into consideration, and so on and so forth. So my only question is that more data does not necessarily translate immediately into more knowledge, but we have a great hope that our colleagues in bioinformatics can help us, because we don't know how to move forward otherwise. Thank you.